let's move on to the next session in the morning here, which is battling soybean cyst nematode in North America, past, present, and future. Obviously more of a concern as we're starting to see more detections in Manitoba. And Greg was supposed to join us a couple of years ago, but his schedule was too hectic. So we've got him here finally, and welcome here, Greg. So Greg Tylka was born and raised in southwestern Pennsylvania near Pittsburgh, which is in the United States, for those of you who were blissfully unaware. He has a bachelor's and a master's degree in biology from California University of Pennsylvania and a PhD degree in plant pathology from the University of Georgia. Greg joined Iowa State University in February of 1990, just a couple of years ago, and he has research and extension responsibilities that focus almost exclusively on the biology and management of the soybean cyst nematode heteroderoglycines, I hope. And if not, Greg, please correct me, and welcome here. Start sharing your screen and dazzle us.
Thank you. That was perfect. Probably too much information about me. So I'm going to jump right to it. I assume, oh, no, there's the first glitch. We're jumping right to it in the middle. Let me go to the front of the presentation.
I hope this doesn't steal your thunder.
Oh, that's all right. Okay. Only done this a few hundred times over the last year or two. So yes, as Tammy said, I wanted to talk about SCN in your country and mine, and talk about managing it, battling it, where we've been, where we are, and where we're going. One of the things I wanted to start with was a map of where SCN has been found. And every 3 to 5 years, we update this map. We just updated it 12 months ago. It was published in January or February of 2021. And the next previous map was 2017. And so I've highlighted on your screen in blue the counties and municipalities that were newly discovered to have SCN in that 3-year period. So kind of some summary stuff on the right side. There were 5 new counties in Ontario, 15 new counties in Quebec, and 4 new municipalities in Manitoba at the time, as well as 55 new counties in the US. Here is a zoomed in look at Manitoba and the 4 municipalities that were highlighted in blue on the map, the previous map. And since that map was put out in January, February of 2021, there's been a new municipality discovered as well. So I think the official, or perhaps the unofficial count of municipalities having soybean cyst nematode in Manitoba is these 5 showing on the screen. And you'll see at the bottom, I've referenced some sources of my information. Mario Tenuta had sent me these maps. So I want to jump right to managing SCN. And as you might expect, it's very similar for you and for me, or for your growers and for my growers. First thing is to don't grow soybeans, as I would say, but my soybean association asked me to rephrase it as growing non-host crops. And so anytime you grow a non-host crop, such as corn in my state, or I took a guess and did some internet searching as to what might be grown up in your part of the world. Anytime you grow a non-host crop, what happens is the eggs, many of them release the juvenile worms. And so here's the egg on the left side, and half of the eggs are just going to hatch when it's warm and moist enough. And these little juveniles are going to starve if they're hatching out in a year that a non-host crop is grown. Now in Iowa, we see the magnitude of decrease in a year of corn as being as little as 5 or 10% to as much as 50% in that first year of corn away from soybeans. I don't know why it's so variable. I blame it on soil conditions and weather, but I blame everything I don't understand and have an explanation for. I blame it on soil conditions and weather. What we do know is if you grow 2nd year or 3rd year corn, in Iowa at least, we see a decreasing decrease. It's a tongue twister, but the rate of decline of the number of eggs the rate of eggs hatching out and the little worms starving is less in second-year corn and even less in third-year corn. And the reason is 40 or 50% of the eggs that SCN produces are dormant, and they're not going to hatch out that first year that they're in the soil or even a second year. So it's the non-dormant eggs hatching out in first-year corn and second-year corn. And by the time you get to third-year corn, there's hardly anything left in the soil except for dormant eggs. And those are going to slowly hatch out over a period of many years. So you'll get a pretty good drop in a year of corn, or I would expect any other non-host crop. But it's a declining return as you grow multiple years of non-host crop, but still numbers will drop every year. Now, to make this a little more geographically pertinent to you all, I also thought about what else might be grown up in Manitoba. And so I want to warn you that edible beans are good hosts for SCN. In fact, some edible beans are better hosts for SCN than soybeans are. And then with the garden peas, the green peas, it's a really weird plant. Some varieties are hosts, good hosts, and others are not. So you'd have to be careful with peas, and you definitely would want to avoid most any type of edible bean because they could raise your SCN numbers. And there's research that shows, not surprisingly, SCN would decrease the yields of those edible beans. The main management strategy and the main topic of my presentation today is cyst-resistant soybeans. And I'm going to finish up with just a tiny bit of information about nematode seed treatments. But the main part of what I have to say the next 10, 15 minutes is going to be about cyst-resistant soybeans. So cyst-resistant soybeans are wonderful. You literally can, just by switching the seed that you plant, go from stunted yellow beans that yield poorly to these very vigorous, tall, green soybeans. And I should interject at this point that the definition of a resistant soybean, such as what you'd see on the right there, is a variety that allows less than 10% reproduction compared to a susceptible. So in a greenhouse study, if a susceptible plant supported 200 females per root, as a susceptible resistant plant would support 20 females or less. And so that's the degree of control or degree of reproduction is measured in the greenhouse, but it translates to performance in the field. So as Tammy had introduced me, I've been around for a while. She was polite and said just a few years. But ever since I've been at Iowa State, pretty much I've started to make lists of SCN resistant varieties. And my first list came out in 1991, and it would fit on the front side of a single sheet of paper. And our list that we just put out in October of 2021 was 24 pages long. And so this bar graph shows you the great increase in selection and choices of varieties that Iowa farmers have to pick from that are SCN resistant. Now, when a soybean breeder wants to make a resistant variety, of course, soybeans aren't naturally resistant. That's why SCN is such a pest. But researchers have discovered these weird soybean lines shown on the screen. We call them sources of resistance. And some of them are from Asia. A lot of them are from Asia. There are 7 main sources of resistance used in breeding programs. And on the right just shows the researchers that published in the year that they published that these were resistant in the literature. Make no mistake, though, that even though these are soybeans, they are horrific when it comes to agronomics. So no one in their right mind would grow pure Peking or pure 88788. But soybean breeders take these and they cross them with good agronomic soybean varieties, and then they select for offspring that maintain those good agronomics, but also have these useful SCN resistance genes that are present in these breeding lines. And in addition to the 7 main ones shown on the screen, I do have written there at the bottom, there are many other breeding lines that have been reported by researchers that have SCN resistance as well. So there's lots of choices for breeding programs to pick from to make resistant soybean varieties. Unfortunately, for my state and for much of the upper United States, the varieties that are available only have resistance from these two sources. And this is what that bar graph I showed you earlier looks like, colored in based on the source of resistance. And so the gray part of the bar represents the number of varieties that have the 88788 resistance, and the red part of each bar has the number of varieties that have any other type of resistance, including that Peking resistance. And obviously, virtually everything that Iowa farmers have available to them have the PI 88788 source of resistance. And the little bit of red part of the bar that represents any other source is almost exclusively Peking resistance. So we're at 95% of our varieties have 88788 resistance, and about 5% have Peking resistance. And an interesting fun fact, or not so fun fact, is that you'd be hard pressed to find a commercial variety to grow in Iowa from a seed company that is described as susceptible to soybean cyst nematode. Almost every new variety that comes on the market has cyst resistance, and primarily 88788 resistance built in. Now, for your situation, it's even more narrow or less diverse. And Mario Tenuta from University of Manitoba and Dennis Lang, who's on this webinar, I want to thank him publicly, both provided me information about approved varieties with SCN resistance for Manitoba. And I know this is hard to capture in a fleeting slide like I'm going to be showing you, but I'll have an email address at the end that you can contact me, and I can send you this list. And again, to reinforce the point there in the text box in the bottom left, that I believe all of this has 88788, but you're building up some choices, at least among the varieties with 88788. And I should interject also that not every variety with the same source of resistance offer the same amount of nematode control or yield potential. The yield potential part is probably intuitive to you all as agronomists, but not every variety with 88788 resistance controls the nematode equally well. Dennis also shared with me some great numbers about acres of soybeans grown in Manitoba and what proportion of those had SCN resistance. So I made a table out of the information he sent me. And the left column is the last 3 years that he has tracked. The middle column is the total acres of soybeans grown in Manitoba. And then the right column is the acres of cyst-resistant beans grown of those total acres. And then I calculated the percentages there in blue. Thanks again, Dennis, for that information. So in Iowa, soybeans are a big deal. Soybean cyst nematode is a big deal. And then reproduction of SCN on resistant soybeans is a big deal. So every experiment we ever conduct in the field, we do a test, a greenhouse test called an HG type test, to see what's the ability of that nematode that lives in that field to reproduce on these main resistance sources. And so we do a test with all 7 of the resistance sources, but we're most interested in Peking and 88788. And again, we do this on every experiment we do in the field. And I should mention that we do field experiments in farmers' fields. We rent from farmers. And so these results tell us kind of what's happening out there in the countryside. And we've done just about 300 of these tests on different SCN populations in 300 different fields that we've worked in. And so I've taken a look back at all those data recently and to see if there were changes in how well PI 88788 and Peking controlled soybean cyst nematode. And what I found is in the decade of the 90s, 88788 resistance worked really well. The level of reproduction, which on this slide we call a female index, but it's that percentage of reproduction I talked about. And remember, effective resistance should be below 10%. We had that with 88788 in the 90s. And another way to look at percent reproduction or female index is that the resistance is providing greater than 90% control. So percent control is just the flip side of how much reproduction it allows. And 88788 worked as it was supposed to in the 90s, but things started changing in the early 2000s. And so these are percent reproduction or female index data, just like I was alluding to in the previous slide. And so each dot on this graph represents the level of reproduction of an SCN population in the field that we rented from a farmer to conduct our experiment. So whatever field this was in 2001 that we rented from Frank Farmer in Farnhamville, Iowa, I'm making those names up, but the SCN population that lived in that field, when we brought it in the greenhouse, it had 28% reproduction on 88788, pure 88788. So again, this shows the trend for all those populations and their level of reproduction on 88788 over the last 22 years. And then I've also put a reference green bar or green line in there. Effective resistance should be at 10% or below. So obviously we see the nematodes that live in the fields in Iowa are building up the ability to reproduce on 88788 resistance. And this is particularly alarming when you think back 5 minutes ago to the bar graph that I showed you that talked about what's the source of resistance that's available in the varieties that Iowa farmers have to pick from. Now, a bit of good news is we look at the same data for Peking, the other source of resistance that's available in Iowa. And in the decade of the 90s, it was almost perfect. There was virtually 0% reproduction of any SCN population from any field in Iowa on Peking. And so again, the other way I present that or have you look at it is that Peking was providing nearly 100% control in the 90s. And then here is a graph identical virtually to the graph previously for 88788. So it's set up the same. Percent reproduction or female index, this time for Peking, and for the last 22 years, from the year 2000 all the way to this current growing season, 2021. And each little red square now in this slide represents the SCN population in a farmer's field where we did an experiment. So this little red square here represents a field we worked in 2004. And the SCN that was in that farmer's field was right at around 10%. And obviously you see a very different trend over time with Peking resistance compared to 88788 resistance. And I guess there's no wonder that this is a much different looking graph than the previous one when you consider the bar graph I showed you, where virtually everything farmers are growing doesn't have Peking resistance, it has 88788 resistance. So here's the two graphs I just showed you side by side to give you a bit of contrast between what's happening in the fields in Iowa with SCN populations as they're able to be controlled by 88788 or Peking. Now this has some real yield consequences. We do variety evaluation experiments at 9 locations each year throughout Iowa. And here's some really telling data from 2019. It's from a location in Southeast Iowa that's really sandy. It's an extreme environment. It's something that Soybean Cyst Nematode loves, sandy soil. And so it represents, I'd say it represents a vision of the future for medium and heavy textured soils. In this experiment, the starting egg count was 4,700 eggs per half a cup of soil. That's kind of the low end of what I call a moderate infestation level. The level of reproduction of the nematode that lived in that field on 88788 was pretty high, and there was virtually no reproduction on Peking in that greenhouse experiment that we did to start the season to test the base nematode that was living in the field. So in this experiment, there were 3 susceptible soybean varieties. There were 68 varieties with PI 88788 resistance, and there were 2 Peking varieties. And this is the yield data. Combined, those 68 PI 88788 varieties, which represent what most farmers have available to grow, they yielded 51.2 bushels per acre. the Peking varieties average 72 bushels per acre. Now, we rent from farmers, I've mentioned that, and we rent about 5 acres, and this field in Fruitland was about 30 acres. That part of the state has really small fields, so 30 acres is small for an Iowa soybean field. But the farmer had 25 acres that he farmed around us, and he grew this soybean variety. It just happened to be in our experiment as well. And he told us that he grew that, and in our studies, the average yield for that variety was 50 bushels per acre. So what I did was take 72.4 bushels per acre. If he had grown a Peking bean, minus 50 is 22.4 bushels per acre. And back in 2019, soybeans were worth $9 a bushel in the US. So he lost out on almost $200 an acre by not having optimum nematode control by growing a Peking resistant variety. And that's per acre. And if you multiply that by the 25 acres he grew that variety around us, he lost out on about $5,000 of yield due to yield loss from SCN because 88788 resistance is losing its effectiveness. And if all that weren't bad enough, these are research plots. We pull soil samples at the beginning and the end of the season to measure nematode numbers. And I just averaged the ending nematode number under the 88788 varieties was 14,400. Under the susceptible, it was 13,600. And under the Peking varieties, it was 800 eggs per half a cup or 100 cc's of soil. That is going to affect soybean yields moving forward as he continues in his corn and soybean rotation. So there is a real consequence to PI 88788 losing its effectiveness. So summary of the cyst-resistant soybean variety information for you and for me. Almost all resistance is 88788. Populations of SCN in Iowa have increased reproduction on 88788. And I asked the rhetorical question, is there any reason this won't happen in Canada eventually? And actually, Albert Tenuta in Ontario, I think maybe Tom Walacki as well from Ontario, have data to show that nematodes in Ontario are building up the ability to reproduce on 88788. From my perspective in Iowa, there's no indication that there's going to be more Peking varieties made available for us or any other non-88788 resistance. I think everything's going to be, almost everything's going to have 88788 resistance. And again, another rhetorical question is there any reason to believe things are going to be different for you all? I kind of don't think so. The last thing I wanted to do was, because we're running out of time, is to just briefly touch on seed treatments. There are lots of different seed treatments available, and they are a great addition to the management toolbox, but they're not a replacement for resistance. And we do lots of field studies on nematode seed treatments as well. And here's just a summary table. We don't have time to go into all the nitty-gritty details, but we've done about 182 experiments in the past 6 or 7 or 8 years. What I have shown here is the different products we've tested, the different years where we did the experiments, and then a summary of the number of times we got a significant yield increase compared to the number of experiments that we tested it in. And then I've calculated kind of a percent frequency of a significant yield increase. And so unfortunately, we don't see a real high frequency of nematode protectant seed treatments increasing yields, but when it does increase yields, the magnitude of that yield increase is almost always economically beneficial. So a seed treatment may cost between $8 and $25 an acre. The current price of soybeans, you can plug in whatever you want, from $9 to $13 a bushel. Your break-even point for a farmer to use this seed treatment, if it provided a yield increase, the yield increase almost always exceeds the break-even point. And so I summarize this really inconsistent situation with a really profound quote, and that is that seed treatments work when they do and don't when they don't. And so unfortunately, I can't predict when we're going to see significant yield increases. But when we do see those yield increases, it makes money for the farmers. So looking forward, managing SCN next year and beyond, my advice to you all for your farmers is continue to grow non-host crops, and the more frequent, the better. Also, they need to grow cyst-resistant beans as we do. We all probably have to use 88788 resistance for now. If you can get a hold of some Peking resistant varieties, those would be great. And then a bit of good news to end on good news. You may or may not know that BASF has approval to develop a transgenic BT SCN resistant variety. In the US, our EPA approved it in the summer of 2020. And then some BASF scientists published this paper in this journal, Nature Communications, just this past June. And if you want to read this paper and read about the BT SCN resistance, you could just Google CRY protein, which is the BT protein, and SCN, and it will pop up. Or you could just contact me directly and I could send you a PDF of the paper. And then finally, I had checked with my contacts, and their plans are to bring this to market both in the United States and Canada. Finally, very last slide, you're also going to want to work with your farmers to use these nematode protectant seed treatments. They're not going to save a crop from SCN, but hopefully they'll top it off with some extra yield. And I want to acknowledge one more time the great information I got from Dennis, who's connected here today, as well as Mario Tenuta, and then my go-to guy up until recently for SCN stuff in Canada was Albert over in Ontario. So they're all three great resource sources for SCN stuff. And with that, whatever time we have left, I'd be happy to entertain questions and comments.
Thank you, Greg. So the first question that has popped up is that when you're looking at that egg hatch, and if there's 50% that hatch in year 1 and year 2 is decreased, if there were only 5 or 10% that hatch in that first year, do you still see a significant decrease in hatch in year 2, or would it be higher? Do you know?
Yeah, that's a great question, and we just don't know. That's based on some field experiments we've conducted, but you know, we do these for 2 or 3 years, these research studies in a limited number of sites, and then we kind of move on to other topics. So it could be better, could be more of a decrease in year 2 or 3, or it could be the same or worse. And there's no real good research papers to help answer that either. I apologize for that.
That's all right. You can do the research and then get back to us. That'll be fine.
Okay. Maybe next year. Yeah.
And just quickly, is Peking doing better because it's currently deployed less frequently?
Yeah, absolutely. The nematode, it's no different than any other control tactic. I call it a force of nature. The speaker prior to me, I think, talked about herbicide resistance. So it's whatever we're using to control a pest, Mother Nature finds a way to overcome that. And so Peking is working better because we've exhausted the effectiveness of 88788. And we'll eventually lose the effectiveness of Peking. We would never want to have farmers switch completely to Peking if they could do that, because we'll go down the same path because of these forces of nature. We want to mix 88788 and Peking and maybe the BT resistance moving forward.
So don't throw the baby out with the bathwater.
Exactly.
Yeah. There we go.
Yeah.
All right. And so then as we're looking forward to the future, and we're talking about, you know, either using the new breeding technique and the introduction of transgenic lines, or if we're looking at other sources of resistance, how are we breeding those in? How is that working? Will we see lots of varieties in the future ramp up on some of those other sources of resistance, or is it super complicated and I don't know how we'll ever get there?
Yeah, so I don't think it's going to happen any quicker in the short term. It's through traditional breeding and back crossing and purifying and so forth. But there's hope because there's new genetic engineering tools called CRISPR-Cas and other things. And once we can hone in on a specific resistance gene for SCN, we can then precisely take it out of a source of resistance and drop it into a soybean variety that's not resistant without doing the traditional breeding. But I think we're probably 5 or 10 years away from, even though we've made great strides, with CRISPR-Cas and other genetic engineering, we're probably 5 or 10 years away from being able to breed new resistant soybeans that efficiently.
And based on some of the other experiences, we know that sometimes there's either a drag of some sort, either a yield drag or maturity, depending on how clean you can snip that piece and introduce it. So yeah, lots of work ahead for everyone. Is that what you're saying? That's
exactly right. That's a good way to summarize.
Perfect. Okay, so I think I've answered all the questions. We're just a minute over time, so I think that that's great. Really appreciate your help and your information there. And I will thank you.